what Euler did. ACR was more hesitant and had a sort of a very hesitant statement originally. But our colleagues from gastroenterology, they were heavily against it because it had not been studied in Crohn's disease. And what EMA and other agencies say, well, if you show us that this is the same molecule with the same properties, the same functional assays, then one study in one indication is okay and you get the rest of the indications. Why should an anti-TNF be different if it's the same molecule, more or less, in the batch-to-batch variation situation? But the ECHO people, the Crohn's group people, made a position statement heavily against having a biosimilar that wasn't studied in their disease. Now, bear in mind, 6 months later, everyone had to use it because the hospitals wouldn't pay for the originator. So that rolled over immediately, more than in the rheumatology field. And they based their critique on the fact that the first or the second biosimilar had a slightly different antibody-mediated cytotoxicity than the originator. Now, you never have the identical things. No protein is the same if it is produced every day. Now, does antibody-mediated cytotoxicity matter for their disease? It doesn't matter really, because what you can see here, that this is an anti-TNF antibody, and you have a lot of ADCC by etanercept, which doesn't work in Crohn's. And you have no ADCC by Trastutsumab because it has no FC, and it works in Crohn's. So ADCC is not important. They had the wrong argument. So no one has ever tested different batches of the originator molecules. I showed you variability, and I'll show you in a minute more variability. So Pekka Kurki, by the way, a rheumatologist working for the agency, wrote a letter back, and he said essentially no pharmacokinetic or safety issues are known to be specific to IBD, and the most responsive population, rheumatoid arthritis, was used to investigate immunogenicity, and therefore the clinical results enabled the extrapolation. And then the Northwich study came, as you know, and they used many diseases where TNF blockers are applied and showed that there was no overall difference between originator and biosimilar. I showed you this slide before, and I said that if manufacturing changes occur in originator molecules, then lots of analytical studies and functional assays are needed, but no clinical trial usually. And so how appropriate is it for the Crohn's people, or was it, now they all use it, for the Crohn's people to accept major manufacturing changes of the originator without demanding trials, but asking for the biosimilar companies to do specific trials, which would make a biosimilar by far more expensive. So let me show you another story. Trastutsumab, breast cancer. What you usually would see is this. In blue, you find the biosimilar. And in orange, you find the originator. So this is what you should be seeing. This is a break-off. But what they saw was in part a different situation. They saw that the originator was worse. And so all of a sudden, they could have been regarded as a bio-better, which would have made a totally different approach to approval. Because they had all the batches, they could show that there was a drift in the relative activity of antibody-dependent cytotoxicity and FC receptor binding. So it was the originator that had the change. And because of the biosimilar study, this was detected. It wouldn't have been detected. And this is what I meant before. How appropriate is it when major manufacturing changes occur to not do a clinical trial to show that it's the same efficacy of the originator as the other one? I think that's an important question. You know, why do I say that's an important question? When we started to discuss biosimilars, the reps of the originator company said, it's not the same, it's not our process, it's not this, it's not that. But they never had told us before that they had changes in the molecule. As you can see here, that as I showed you with etanercept, I could show you with rituximab. So this is what made me a little bit surprised. All of a sudden, because biosimilars are coming up, manufacturing differences are discussed, but obviously only for the other companies, not for your own product. And this is simply not fair. And we should have been told better. The regulators knew. I showed you the ICH 2004, so they had discussed it already long before. I'll show you one of my studies with the Sandoz biosimilar rituximab. So here we studied GP 2030 and the European and the US rituximab, and you can see that the ESTA score is essentially the same all over the place. But look at this. When we looked at B-cell repopulation, what we saw was a major difference between the originators, between the rituximab from Europe and the rituximab from the US, and the biosimilar was right in between. It didn't matter clinically, but it could have mattered clinically. And this is the overall efficacy for all, and you see it's essentially identical. What about anti-drug antibodies? Everyone says, well, the biosimilar will have anti-drug antibodies because it's not produced by the original company. The difference was between Europe and the US, 21%, 13%, overall 15%. The same or is very similar to what was seen with the biosimilar. Now, biosimilars are interesting for the society and for every field, but also for the society because of cost reasons. And you see that some places in Europe take up rapidly. You know what? It's the richest ones. Denmark, Sweden, and Norway. Germany, slow, but finally it does it. Who are the lowest? Slovenia, Poland, this is surprisingly. They should be more interested. This is infliximab biosimilars, that same story. Denmark was the first, and I believe, what is it here? I believe Italy is, interestingly enough, Norway, and then Italy is the next. So what are the cost reductions due to biosimilars for the hospitals, the societies, the field? Why am I interested in cost reductions? Not only because of our societal conscience, our consciousness about society is important. When you go to the grocery store and you see two very nice oranges and you don't see a difference between the oranges, you will probably take the cheaper one, especially if it comes from Greece. You wouldn't take the Italian one that is more expensive. So I mentioned Denmark, and in 2017, the approval was in 2013, 84% of the etanercept was biosimilar. And look at this. So here you have, first of all, this is the originator in dark, so that dropped more or less rapidly. And overall, what you see is an increase in the use of etanercept. But at the same time, you have almost half of the costs. And what about another, infliximab? Same story. Here, there is even a disappearance of infliximab originator from the market because they at least lowered the price. Merck didn't want to lower the price, and that's, I told them, they were with me. They told me, well, why don't you help us? Because we have given lots of deduction to the cardiology drugs. So I said, I don't have to defend cardiology money. I have to defend rheumatology money. So drop it by 20, 30%, and I will not change the therapy of my patients. because the first ones were 30% lower. Now they are all 50% lower by law in my country. But what happened? That's the next story. Again, what you saw here, but even more. Look, infliximab use doubled over time. And this is when the second, this is the first, this is the second infliximab biosimilar. Look, and the costs were more than halved, despite the fact that it was double. So you essentially, per patient, it was 1/3 to 1/4 of costs. More people could get it. Now, if you take less affluent countries, poor countries, they should be interested in this because they can treat more people. I will come back to one story which makes me smile. But what about switching between originator and biosimilar? I know that some people say, well, I can think of using my anti-TNF as from the biosimilar, but my patients like my infliximab or their etanercept or their rituximab, and they don't want to switch. What about switching between originator? Bear in mind, as I implied, we all have already switched to not originators, different batches. And we don't know whether or not these batches had sort of, in some sense, slightly less efficacy or whatever. I showed you an example with Trastutsumab. So here is a study where adalimumab was compared with this is adalimumab, reference adalimumab with biosimilar, and then after week 16, there was flip-flop switch in both arms. And as you will see here, no difference. First of all, no difference for the first 4 months, and then essentially you see here for a year, no difference in the 4 arms. They started slightly, at the end of the day, they get the regression to the mean because they are similar. What about more details? Is PASI, this is psoriasis, by the way. PASI 50 and PASI 90, you can see that initially the symptom, this is after switch, and you see essentially the same. Another psoriasis study with etanercept. Here, again, etanercept versus, this is the reference, this is the Sandoz etanercept. Here are the arms, and they are continued, but then there is flip-flop. And you can see again for the PASI 90, initially the same, and then with the flip-flop, whenever curves, to me, I'm not a statistician. I don't understand much about statistics, but I like to ask questions about statistics. So whenever I see differences like this, I worry. I wonder what that means. However, when I see crisscross, then it's a play of chance. Because here, it's the other way around. And all of a sudden, here it's the same. So you see crisscrosses or identical curves. This is an RA study. Same story or similar story. Compare the Sandoz adalimumab with the reference adalimumab for 6 months and then switch the reference to the biosimilar. And you will see here, this is the change in DAS28 CRP. You will see here for the first 24 weeks, it's more or less superimposable. But then after the switch, there is also no difference, no loss of efficacy. By the way, adalimumab is, among all the proteins that I saw, the batch-to-batch variation has the lowest fluctuation. Now, there is one more problem, and that is, even if I would like to use the biosimilar because my hospital or my insurance wants me to use it. You know what happened to us once? After etanercept biosimilar was already approved, one of my colleagues in the clinic prescribed the originator. So he got a phone call from the payer, from the insurance, Social Security. And they said, Dr. XYZ, do you have a specific reason why you prescribe the originator and not the less expensive biosimilar? If you have a good reason, tell us and we will give it to the patient. He had no good reason. because just believes, that's not a good reason. But very frequently the patients are reluctant, and that is the question. There's two possibilities. One is the patient doesn't want. Well, then you can tell the patient, you know what, if you want to keep that drug, you would have to pay for yourself. Then they very rapidly want it. There's no all of a sudden they don't have a problem to switch. But that's not the right way. It's just what happens, because if the payer doesn't pay, then the patient has to pay themselves for a drug. But the more important thing is to explain to the patients what we discuss here today. We call it framing, positive or negative framing. Mrs. Miller, there is a new drug out there. You know, it's not from the same company. I don't even know if it works the same way. Do you think we should give it to you? Then Mrs. Miller will say, no. But you can say it differently. Mrs. Miller, there is a new drug out there that's the same as your old drug. It just has a different name. And you know what? Over the years, there were many changes in this drug, and we accepted all these changes, and that's just a different batch, and it happens to make it also cheaper for you and the society. So we call it positive or negative framing. And you can see here from this study that the positive framing allows the switching much more frequently than the negative framing. And why is that? Because, and this is work from Italy, this is work from Fabrizio De Benedetti, who looked at MRI in placebo and in nocebo. That's a nocebo effect, because you don't change the treatment. And he found out that a positive expectation with a positive communication activates opioids, endogenous opioids, and endocannabinoids. You know, we are not addicted, all of us, but we know that opioids can make you high, can make you happy, especially the endogenous. Negative communication activates cyclooxygenase and cholecystokinin, and that makes you being afraid. If you want to read more, several years ago, 3 years ago, we have published a consensus-based recommendation for the use of biosimilars to treat rheumatological diseases, a large task force. And there is one more thing that's interesting. I don't know if you heard that. 2 days ago, the FDA gave a warning. Now, I use JAK inhibitors a lot, but the FDA gave a warning. I'm not sure that EMA will also do that. But the FDA said that these medicines, all the JAK inhibitors, should be reserved for patients who have had an inadequate response or intolerance to one or more TNF blockers. They explicitly said TNF blockers. I don't know why. I don't know why they didn't say Alzheimer's. So I'm just bringing this up. If you didn't hear about it, you will hear about it. I don't think EMA will stipulate the same thing, but I don't know. I don't know what the story is in detail, because I haven't seen all the data, and we have registries where the JAK inhibitors behave differently than in the one study, 1133, that elicited all this. But what I wanted to say here is that when they speak of TNF blockers, then also in the United States, there are biosimilars available. Let me close by looking again to the World Medicine Situation Report that comes out every 10 years, so I expect a new one. I mentioned to you that they have 3 requirements. Meet appropriate medicines in doses that meet their requirements for an adequate period of time and at the lowest cost both to the community. And then they say irrational or inappropriate, improper, incorrect use is when one or more of these conditions is not met. And one of them is costs. And I think we all want to see innovative drugs. We all want to use new drugs for our patients. We're happy that the pharmaceutical industry produces new drugs for our patients. But the old drugs, when the originators don't drop, by the way, adalimumab originator made a tender in Norway because they were cheaper than the biosimilars. So, you know, this is what the government in Norway then decided, to continue using the adalimumab biosimilar, the adalimumab originator. But it's a question of price. So let me sum up, Mr. Chairman and colleagues. All BDMARDs have similar efficacy. I didn't show you that, but this is what we have seen. And JAK inhibitors are the least efficacious biologics and show efficacy across many IMiDs. BDMARDs should be combined with methotrexate to convey optimal results. The T2T strategy is target attainment at 6 months and leads to optimal outcomes. But then, biosimilars approved in highly regulated areas, such as in Europe, are as efficacious and safe as originators, and switching from original to biosimilar or between approved biosimilars is like using a different batch, such as due to manufacturing changes of the originator. Originators have not, or very rarely, performed comparative trials between batches of their BDMARDs. But overall, the prospects for RA patients become better with every year. We have good therapies, new therapies, good treatment strategies, and there is more to come. And with that, I'm happy to entertain your questions.
Thank you very much. So the distinguished speaker's lecture is open for discussion. I would like to remind you that this is a satellite symposium supported by Enorasis. And we can open this discussion with questions. Please, no comments, only questions. Is there anyone that has a question for Prof. Smollen? Let me start, Joseph, by asking you something that we see in our clinical practice. Switching. For you, it's very clear. All over the years, through your experience, you stated that, in a sense, the originator is probably not an originator. There is a change in the batch. So the biosimilar and originator, in a sense, are the same thing based on what you have said. Can we translate that in our clinical practice? So for a practicing physician, a junior rheumatologist that has in front of him a patient that has, in a sense, to get either the originator or biosimilar A or biosimilar B. If you are a junior...
We have done this and we have seen no difference, and we explain this to the patients. You know, the only thing that sometimes patients may complain is that the device is slightly different. The injection device. But usually, bear in mind that most of the biosimilar etanercept compounds had less adverse events in the skin, less burning sensation than the originator. That was interesting. Now, that's also interesting. I forgot to mention that. EMA would regard a biosimilar that's safer not as a bio-better. They would only regard a biosimilar that is more efficacious as a bio-better. So when you see, I don't know what it was, 15% versus 5% injection site reactions, I believe in some studies it was even significant statistically. For EMA, that's fine. That's because it's not a major issue of the molecule, but of the packaging of the drug, of the additional compounds.
And what about multiple switching? Because in some countries, we don't have strict regulators. So we may see in our practice switching from an originator to a biosimilar, or from a biosimilar A to a biosimilar B.
Yeah, no problem. It's the same. You did it already with the originator. I showed you 50 manufacturing changes for infliximab, 20 or so for rituximab. We did that because the next batch doesn't go back to the, it's again some difference again. And so it's essentially we have to look at this in our place. Not if you look at, I don't know what, Reditux from India or Isaipu from China. But in our Europe, I think EMA does a wonderful job. I'm always surprised about the logic that is behind the decision of EMA. And I'm not always so seeing the same in logic in the FDA. But EMA does a wonderful job. I'm surprised. I always thought the FDA was better, but I think EMA does a wonderful job. And EMA when EMA, to me, it looks until now, whatever EMA approves is fair and is a good drug. Sometimes they give caveats. That's fine. That's allowed. And they say it's conditional approval. But no, I have no problem switching from one biosimilar to another, and we've done that. You know what? It's not even anymore our prime decision, because it's usually the payer that decides that. And bear in mind that when you do a tender system in Norway and you get an adalimumab biosimilar to be the drug that wins the tender, then for a year the system uses the adalimumab. It doesn't continue. It wouldn't go to the etanercept from adalimumab, but they would still switch the patients, and then the next time another biosimilar C or D could win the tender, and then the patients would have to switch. And I've not heard from my friends in Norway that there is major problems with the patients.
Which brings me to the next question. What about the perception of the patient? Because maybe in Denmark or in Norway or in Austria, the patients don't have a problem from switching every year because of the pending of the insurance from one biosimilar to another biosimilar. But in Mediterranean countries, and in some countries that they're very enthusiastic and they don't like a lot of changes and they are not so very much confident about switching, there is a lot of difficulties in perceiving this kind of switch. It's a matter of education of the patient.
You have a very good point. Well, first of all, about my country, we don't have a tender system. The hospitals some hospitals may have their own tender systems, but by law, the first biosimilar, by law, Parliament decided that. The first biosimilar has, I believe, to come down 35 or 38% below the price of the originator, the last price. The second biosimilar has to come, I believe, 46%. And when the third biosimilar comes from the same compound, then all of them, also the originator, are forced by law to come down to minus 53%. But then the battle starts, because the 53% is the law. And then they want to say that if everything is 53%, they're not going to, who is going, then you can toss a coin. But then they start negotiating, one gives 55%, the other one gives 57%, whatever. Then they start negotiating with the system. But otherwise, if everything stays on 53%, again, you are free to say, well, I want to continue it. My originator, etanercept, I want to do this, I want to do that. So this is only a price issue. Otherwise, I don't see any reason to be worried. It's essentially like with the generic drugs. It's just proteins, but it's essentially like with the generic drugs, where you also don't stick to the very same compound, but very frequently the pharmacists decide what to give the patient.
I will give the microphone to Professor Djoufas for his next question.
This is the Greek orange, Joseph. Sure.
Pleasure to see you.
Pleasure
Thanasi.
to see you too. Pleasure to see you too. Are you convinced?
Can you take off your mask, because I think
sure,
that
sure, sure. I'm sorry. No. I'm going with my mask.
No.
Even
we all carry the mask,
doing shower. I'm sorry.
But,
it's okay, right.
But
Are you
gives you a
convinced
reflection
that different batches or biosimilars, and particularly the different post-translational modifications they get, expectedly, affect or not the immunogenicity of the molecule? This is particularly important, as you know, because Professor Smolin is a great immunologist as well. Are you convinced that this continuous switching might affect the B-cell repertoire towards this?
When you read, this is a very good point. We weren't sure about that, because when you read our consensus paper for 3 or 4 years ago, we say this, that multiple switches should be followed in registries, also potentially with respect to antigenicity, to immunogenicity. Now, on the other hand, we have done for 20 years multiple switches. When an infliximab molecule changes, when an etanercept molecule gets this and has a different glycosylation, and the next time it has another, then we've done this already. We have already had this, but we have never, that's why we said it in the consensus paper, we've never studied it systematically. Now we can study it systematically, but I wouldn't be worried about switches and multiple switches and back and forth switches. So I think that this is fair enough.
Good. Thank you.
Yes, but then what about whether it is a nocebo effect in case that we don't have efficacious, we don't have response?
Yes.
We don't have very nice studies. I'm going to start from the point, it's going to be an immunogenicity, a nocebo.
I agree with you. I couldn't agree more. However, when you take registry data, you will see that there is a drop in efficacy by 10 to 15% every year. Patients stop drugs. The registries show very clearly that infliximab after 5 years, only 50% of the patients have it. So, you know, you first would have to show that this effect goes beyond the natural course of the originator, which it doesn't. Yes, you have seen that if the patients aren't aware what they get, there is the same effect in psoriasis and rheumatoid arthritis. There's a straight line in the Admira study when you switch from adalimumab to the originator to the biosimilar. There's a straight line in the DAS28. There is no drop whatsoever. Whether or not an individual patient can have a reduction, this is the mean. Yes, but they may have a reduction. They probably have the same reduction for the originator. And the registers show us the curves are like that. For the originators, we can look back 20 years of registries. They all show the same. And then they compare, one drug has a better retention rate, and then the other drug, they compare this, and yes, not infrequently, drugs like infliximab, which are more immunogenic, have less retention rates than drugs like etanercept that are less immunogenic. But at the end of the day, when I look at the overall gestalt of data, I didn't see differences between the highly immunogenic adalimumab and the less immunogenic etanercept when they were studied more or less in comparative way. And I myself was the first author of Trastutsumab against adalimumab, head-to-head. Same outcome. Even though Trastutsumab is a humanized antibody fragment, but it's presumably more immunogenic.
Any other question? If there is no other question, then I think that we need to finalize the distinguished lecture session here. We are really privileged that you have around, and I hope that you will stay more next time that you come in
Next
Greece.
year we had planned for this year too, but you know, it didn't happen because it came up. Okay, anyway, but thank you very much, and thanks for the great discussion.
Okay, great. Again, we would like to thank Professor Smolin and, of course, Enorasis for this satellite lecture. Have a nice stay.